chromatography and its types. So these are the contents we are going to learn today. Chromatography and its history, principles, applications, types, paper chromatography. These are types of chromatographies: papers, thin layer and gas liquid. Other chromatography types are there, but in this video we are going to learn about only these three types of. Okay, what is the meaning of chromatography and what is it? So chromatography is actually a physical process. In here, the mixture of the solvents are separated and it will result to given a differential distribution between the stationary and the mobile phases. So actually the chromo, the word, it is a Greek word, it is meaning for color, and graphene is a mean for writing. So the history, it is actually invented by, this technique is invented by Stuart. So he was a Russian scientist. He was referred as his father of chromatography. He was only created for the developed the chromatographic techniques. He continued to work with the chromatography in the first decades for the 20th century. So primarily he was used to the plant pigments like chlorophyll, carotene and xanthophylls. Since these components are having the different colors, green color, orange, yellow, so that's only the technique is known as chromatography. As we already said it, so chromo means the color. So only this technique is known as chromatography. What is the principle based on this chromatography? So chromatography is usually based on the principle of partitions of solutes within two phases. So in here two phases are here. First one is mobile phase and another one is stationary phase. So in mobile phase, in the sense, it is usually referred to mixture of the substances to separated, dissolve in the liquid, or it may be in the gases. In stationary, in the sense, it is the porous solid matrix. It is through the samples containing the mobile phase is percolated. What is the applications? Why it is used? So the chromatography techniques is used for the separation of amino acids, proteins, carbohydrates, and it is also used for the analysis of drugs, hormones and vitamins. Helpful for the qualitative and quantitative analysis of the complex mixtures too. This technique is also used to determine the molecules of proteins. What are the types of chromatography? So here a lot of the types of here, paper chromatography, thin layer chromatography, gas liquid chromatography, column, ion exchange, gas liquid chromatography. Likewise, there is most type of the chromatographies are here. So first one, paper chromatography. So paper chromatography, in the sense, in here the strip of the paper, that means the chromatography paper is used as the one. So in here the paper chromatography is the technique, a small dot or the line of the sample solution was kept in the bottom of the paper, and this paper is kept inside the jar which containing the solvents, and it will be sealed. So after that it will be kept in the undisturbed one. So as the solvent rises through the paper, it meets the mixture of the samples where we are spotted, so which started to travel along with the solvents too. So this is the one. So different components in the sample mixtures are travel along with the solvents. It will be given at different rates. It may be taken as several minutes to several hours too. After that one, that is analysis by the spots which corresponding to the different components. Maybe it is located by the color or UV light or ninhydrin while we are spraying the ninhydrin sprays, or by treating with iodine papers like that. So in here the R of value, it was calculated by the R of means, that is a distance traveled by the substances. It is divided by distance traveled by the solvent. Okay, in here there is two type of the chromatography here. First one is ascending and another one is descending. The procedure is the same as the paper chromatography, but the travel, the direction only, it is only different in here. So ascending means in here it is from the bottom to the up. it will rise up from the what will happen? It will from the bottom to up, it will move ascending order. In descending, it is the vice versa. It is move from top to flow. So in the ascending one, the solvents are kept in the bottom of the jars, and in descending one, it is the solvents are kept in the top of the chambers. So that only it will move along with the mixtures of the samples are move along with the solvents, then it will be spotted and analyzes. Significance of the paper chromatography. So it is very easy and simple, rapid and highly efficient method for separations. It can be applied in even microgram quantities is enough for the analysis, and it can be used for the separations of wide variety of materials like amino acids. We already seen it in amino acids, oligosaccharides, glycosides, purines and pyrimidines, steroids, vitamins, alkaloids like penicillin, tetracycline and streptomycin. The next type is thin layer chromatography. In here it is also the chromatography, paper chromatography only, but in here the stationary, instead of using the stationary phase of the paper, here we are using as a thin layer of absorptions like silica gel, alumina powder, or it may be cellulose. They are instead of used as a paper. And to compare to the paper, it is advantages of fast runs, better separations and the choice between the absorptions. So these are the types. So this is a jar and this is the thin layer plate, and here the absorptions are the blue one, it is the absorptions of the silica powder or any other ones. So here we spotted, and it will the same as the technical principle like a paper chromatography. Okay, the next one is the gas liquid chromatography. So here it is also the same thing, but here the solvents, instead of the solvents, here the mobile phases act as a gas. So in this method, the choice of separation of the volatile substances or the volatile derivatives or certain non-volatile substances are here is used. The stationary phases are in here the inert solid materials impregnated in the non-volatile liquids. So this gas chromatography, or it is maybe otherwise known as gas liquid chromatography too. So in the methods, in the sense, so in gas chromatography, a sample of rapids heatly and vaporized at the injected port, and here the samples are transported through the column at the mobile phase consists of the inert gas, and here the based on the boiling point of the one, it will separated, and it will relative affinity, maybe the boiling point or it may be the relative affinity. Based on this, it will be separated. And here the most often a liquid is within the column. The higher component affinity of the stationary phase is the lower comes of the column. So the components are then directed and the represent of the peaks of the chromatography. Okay. So the mixture of the volatile materials injected to the columns along with the mobile phase, and the separation techniques. In this separation, the volatile mixtures is based on the partition of the components between mobile phase. It may be gas, and the another one, the stationary phase, it is the liquid. So here we are using the gas as well as the liquid. So that's only this name is known as gas liquid chromatography. Here the uses are, it is well suited for use in the petrochemicals, environmental monitoring and the industrial chemical fields. And it is sensitive, but it is rapid and reliable. So up to this I conclude my video. The next types and the methods will be continued in the next video. Thank you.